because we are refocusing on our chapter development, and we want to get more people involved. And that doesn't just mean the people that are affected by blindness, but it's everybody else in the community. The goal for me is to make the Foundation Fighting Blindness just a common word that everybody knows. It's like, cancer, leukemia, and all those other groups that are out there, and we need to be just as recognized as anybody else out there. And so that's why this is very important to have the speaker series, because I know people are interested in the research that we're doing. What kind of funds are we generating for the different diseases and things like that? And I know Allie, she'll be talking a little bit more on that, but in general, I mean, the whole upper management at the foundation has changed for me in the last 2 years. So I'm actually learning new people as we go. But for all of you new people that are in today's meeting, I should probably introduce myself a little further. Again, I'm Rick Dahlstrom. I am affected with a disease called choroideremia. It's very similar to retinitis pigmentosa in the fact that I'm losing my vision from the outside in. So I'm essentially in a state of tunnel vision these days. I don't have any night vision. My peripheral vision is very poor. And this has been going on since I was at the age of 8, and right now I'm 54 years old. So it's something that's been progressive. I've known it's been coming. It hasn't been something that's taken me by surprise, but here recently, it's something that's affecting my lifestyle more. I mean, I found that in my 30s, I was slowly not being able to compensate for my lack of vision. And my frustration levels were growing and things like that. And so now I'm at the point where, I haven't driven a car other than pulling it in and out of the garage for about 4 years. I am just now making my inquiries to the league and Fred and his group to possibly start cane training fairly soon, because I'm finding that I'm needing the assistance. I learned a long time ago, even though it's difficult to ask for help, and that's what the league is here for. And I'm glad we are teaming up with the league finally, because they are a crucial part of the Fort Wayne community. And one of the things that it still is going to be a concern for me is I am a practicing architect. I'm slowly changing my work description. I don't go out in the field anymore. It's just too dangerous. But I'm finding that I have other crucial things to do in the office and help mentoring the new architects, things like that. So I'm trying to adapt. I mean, I guess that's the key part. And there's people that are, have been supporting me all the way through, and I'm so proud to have all of you here with us today. Trying to think where else I want to go. But again, that's my spiel. If anybody wants to contact me or for any reason, don't hesitate. My information's on the foundation's website and things like that. And most of you know, there's quite a few people with my information now, so that's okay. And with that, I think I will introduce Ali Sarwar. She is our, the titles keep changing. She is our chapter engagement manager. So this is a little different spin on things, but Ali is here to support the chapter. This is not to say that Linda Shellis is out of the picture. Linda is still a part of the Vision Walk. Oh, that's what I wanted to say. I do want to give a shout out to those of you who have been supporting the Vision Walk. You guys have been doing a great job, and um, even though we didn't meet our markers these last 2 years, I can't ask for more. I mean, that's just where I'm at. So thank you very much. And Allie, it's all yours.
Yes, I'm glad you mentioned Linda Shellish. So she's the Vision Walk manager for Fort Wayne, and she is actually at Twin Cities right now doing a Vision Walk. So that's why she can't be a part of this call. But thank you, Rick, for going over your journey with the foundation and our chapter developments. I want to let you guys know I will be sharing my screen. Let's see. There we go. Okay. So as you guys know, this is the Fort Wayne chapter speaker series, but my main focus today, what I'm going to be going over, actually, before I get started, I can't see head nods or anything, but a verbal yes. Can you all see that I'm sharing my screen, or there's a PowerPoint up?
Yes.
Okay, awesome. Thank you. So the main thing I want to go over today is foundation highlights and research advancements. So starting with that is our track record. And the Foundation Fighting Blindness is the world's leading organization committed to finding treatments and cures for retinal diseases. And we're pretty proud of being able to say that. Going off of that, as of today, we've raised over 816 million in terms of raising and advocating, supporting research advancements that will reverse blindness and restore vision since our inception in 1971. So part of that, last year, to give you a little more idea, the foundation received 82 grants, and this includes over 90 investigators and 67 institutions, eye hospitals, and universities. And we've also funded studies at hundreds of prominent institutions throughout the world. So that's where our global funding comes in. To give you a few examples, Wilmer Eye Institute from Johns Hopkins University School of Medicine, Massachusetts Eye and Ear Infirmary, Harvard Medical School, Institut de la Vision in Paris, France, Moorfields Eye Hospital, University College London, and also Shea Eye Institute, University of Pennsylvania. So through all of that, providing that funding, there's been breakthroughs, which includes Luxturna. I'm sure a lot of you may be familiar with that. It's our first gene therapy for the eye. And then also Argus 2, which is an FDA approved retinal prosthesis for a bionicle retina. So just through all of that, we're really excited to get the momentum going. And, I'm excited. I keep needing to update these statistics for each of the presentations that I have. So next is our inherited retinal disease landscape. So over 200,000 individuals in the US have a rare inherited retinal disease. And there's a typo in this, which I need to let our marketing know. But, and that includes 4.5 million globally. So we want to make sure that we can find a treatment for everyone. Also, over 10 million people in the US are affected with age-related macular degeneration. And globally, that's actually 150 million. Lastly, we have 45 clinical trials underway or being prepared to launch for potential treatments to help those individuals. The next slide that I'm going over kind of goes more in depth. It's identifying inherited retinal diseases, and it shows that so far we've identified 270 genes that can cause retinal disease. And to give you more of a breakdown, 65 of those genes are retinitis pigmentosa. For Stargardt disease, it's 21 genes. For liberal congenital amaurosis, it's 14 genes. And then for Usher syndrome, it's 15 genes. And, we are studying other retinal diseases as well, but those are the main ones that are identified. The next slide I'm going over has a lot going on. So it just goes over our signature programs and initiatives. And the first one that I obviously want to bring up is our chapters. So we have over 40 volunteer-led chapters. So it's a national network that raises funds, increases public awareness, and provides support to the community. And this is where Rick Dahlstrom, Vicki Western are speaking, because they're a part of our chapter leadership team, and it continues to grow. The fact that we're having the speaker series today is a part of the chapters program. So we're really excited to have that development and continue to grow Fort Wayne. And then there's the clinical consortium, which is, we have 20 plus clinical centers of excellence with experience in IRDs with standardized assessment protocols. Then we have our RD fund, which was launched in the end of 2018, where the Retinal Disease Venture Philanthropy Fund drives emerging therapies that are moving towards or in clinical trials. And I understand a lot of this might be not, maybe a little too much information, but I want you guys to understand how much the foundation is doing. So going off of that, there's actually 6 more programs and initiatives. My Retina tracker, that's a huge one. I want to make sure if you're affected or has a loved one that's affected, please make sure you're signed up for that. We have a patient registry with over 10,000 profiles. It's global, it's free, it's secure. So if you have any questions, please reach out to me about that. Well, any of this, please feel free to reach out to me, and I can make sure you're in contact with the right person. Then we have grants and awards. So that supports the clinical trials, the preclinical research applicable to a broad range of retinal degenerative diseases. And as Rick has mentioned, Fort Wayne has a vision walk, and there's actually 35 other vision walks throughout the nation. So that's our main fundraiser, I would say. And we've raised more than 58 million since spring 2006, which was our first vision walk. Then we have our dinners, galas, and events. So we have dining in the dark, we have visions ball for certain areas. And then also, I think events that kind of goes into DIY fundraisers, so do-it-yourself fundraisers. So really anything that someone's interested in putting together that they would like to go towards the foundation. So anything to raise awareness and research funding. And then visions, which last time we've had it, that was virtual, but it's the foundation providing a supportive learning environment through conferences, conference seminars, and workshops. So it's similar to the speaker series that we have today, but it's typically in person, just like we would hope to have our next speaker series to be, but it's a panel and it's a 2-day conference. So it's a little more in depth. And then lastly, we have BioBonds, which is an advocacy program, and it's about the long-term opportunities for advancing new students' studies and for Biomedical Research Act. So if you are interested in doing that, we have a template of what you can send to your local representative. But other than that, I think I will, actually, no, I want to do one more screen, because I want to thank our national partners. So these are individuals that give thousands and thousands of dollars in support of the Foundation Fighting Blindness. So starting off with Genentech, Janssen, part of pharmaceutical companies of Johnson & Johnson, and then AGTC, Adidas Medicine, Apellis, Astellas, Biogen, Mira GTX, and Spark Therapeutics. And with all that said, I know that was a bunch of information. This is being recorded as, I'm sorry that Rick, during your speech that you were interrupted with that. This is being recorded statement, but I want to make sure that you all can review that. And if you have any questions, please feel free to put it in the chat or ask away now. But I think I'm going to go ahead and hand it off to Vicki. Oh, hold on, Vicki, I need to unmute you, apparently. There we go.
There. Okay. Well, thank you, Allie. I appreciate everything the foundation's been doing for us, and especially starting this local chapter up again. So today I want to introduce Dr. Leung. He studies retinal degeneration, which is a group of incurable eye diseases, including retinitis pigmentosa and age-related macular degeneration that affects vision. He currently hails from Purdue University, where he's an associate professor in the Department of Biological Sciences. So let's welcome Dr. Leung.
Thank you very much, Vicky, and everyone for giving me a chance to share with you what I have been doing. Since I was a young student, I was a student from Hong Kong. These days, if you hear Hong Kong from the news, it's not going to be good news. But anyway, I'm from Hong Kong. I've been studying eye diseases since 1998. I study different types and use different skills. And today I'm going to share with you some of the recent things that we have been doing at Purdue in the last few years. So first, let me share the screen, and then I will continue to tell you the story.
Do you see a share screen button?
I see.
Okay,
I just do have quite a few windows on my screen. I need to find the right one.
That's exactly what I went through too. Okay.
Right. Can everyone see my screen? Well, what I'm going to do is I'm going to make the pictures and the fonts very large. And I'm also going to talk very slowly. So I understand some of us may not be able to see the pictures very well. And I'm going to tell you the story, even though if you cannot see well, you can still get the information.
Right now it's just a
black screen. Right now,
okay, good. Okay, good.
Screen. And today I'm going to tell you a story related to the zebrafish, which is a fish that you can find in the pet store. In the lab, we use this animal model to study eye diseases. And I'm sure you are very familiar with these eye diseases that can affect our good vision. Of course, there are many types. And today I'm going to focus on one of them, which is the retinitis pigmentosa, and also commonly known as the night blindness. In these patients, their rod photoreceptors are affected. And photoreceptors are the neurons in our eyes that are responsible for picking up the light from the environment. And when the rod photoreceptor degenerate, and we begin to lose our vision. And in humans, when you lose the rods, I'm going to bring up a laser pointer, you will tend to lose the peripheral vision, or you are left with a tunnel vision. Because in our retina, the rod photoreceptors are mainly found in the periphery. And in the center, where the part where we used to read and do other things, that part has a lot of cone photoreceptors. And so these are the two types of photoreceptors we have. Rods are for dim light and peripheral vision, and cones are for the bright light and color and acute vision. In our lab, we use the fish as the model to study the eye diseases. We use the model to find out why the retina or the eye is falling apart. We study the disease mechanism. And at the same time, we use the animal model to discover new possible drugs that can be used for treating patients. These two research directions cross-pollinate with each other, and they help each other. And then in the end, the information that we find, we would like to bring that back to the clinic and help people who are suffering from the eye diseases. And today we will spend a little more effort to tell you what we have been doing and finding in the drug discovery direction. I'm going to separate my short talk into 3 parts today. First, I'm going to tell you a little bit about zebrafish, this animal model, why we like to use that to do research, and why many of us in the eye research field like to use it to do our studies. And then I'm going to tell you one of the ways that I use in the lab to test the vision of the eye disease zebrafish. And then at the end of my talk, I'm going to tell you how we are going to use this platform to screen and identify new drugs that we can use to treat eye diseases. And in particular, today's example would be the night blindness, retinitis pigmentosa. So now I'm going to tell you a few things, a few advantages about the fish model that we like it very much for our research. The next picture that I'm showing on the screen shows you the first advantage of the fish. In the center, we have an adult zebrafish, which is not very big. And we can keep a lot of them in the research lab. And when we keep them well, feed them good food, and they can be very prolific. Each pair of adult fish can lay up to 100 to 200 eggs every week. So when we do research, we do experiments, and we want to do that many times so that we are sure and confident about our observations. And when we do experiments on the fish embryos, we can do that repeatedly because we can collect hundreds, if not thousands of eggs in a short period of time. So that helps us to increase our confidence of the finding. At the same time, when we use fish embryos to do our research, there is another advantage, which is shown on this picture on the slide, which is the fish embryos develop very fast. From the time that the egg is born to the time that a little fish has grown to the shape that you can recognize, you only need about like 24 hours. In just one day, you start to see a little fish in the container. And that also includes the eye. And in fact, Mother Nature really forced the little fish to develop vision very quickly, because in the wild, the fish embryos are swimming in shallow river. And then if they don't develop vision and escape from the predators, they will become someone's lunch. So in 2-3 days' time, the little zebrafish begin to see the world. And when we are using these embryos to study vision and study eye diseases, we can collect a lot of embryos and they start to see very quickly. So that helps us to do a lot of experiment in short successions. Not only that, the fish eye, starting from the embryo, they share a lot of similarities with human eyes and retinas. What I am showing on the screen is a histological section of a 5-day-old zebrafish eye. Histology is a way for us to cut the animal open and look at the cellular structure. I would like to draw your attention to the center, the blue part of the eyeball, where we find as the retina of the fish. Just like humans, fish retina has 3 cellular layers: the photoreceptor, the inner nuclear layer, and the ganglion cell layer. We don't need to go into the details, but the bottom line is we have the same 3 cellular layers, and we find the rods and cones in the photoreceptor layer. Just like humans, these cells are connected to each other in 2 structures or layers called outer plexiform and inner plexiform layer. And also very like us, the retina finally will convey the light signal to the brain through the optic nerve. On our left-hand side, we have a blue ball, and that is the fish lens. So we also have a lens. The black layer here on your right, or closer to the center, is the retinal pigment epithelium that humans also have. And it is responsible for a lot of well-being and physiology of the retina and the photoreceptors. So by showing you these, I'm not asking you to memorize anything. This is not an undergraduate course, but I'm going to emphasize that the structure of the zebrafish eye is very similar to the ones that we have. And the consequence is things that we learn from zebrafish research can potentially be applicable to human use. So we have talked about quite a few advantages of the fish model. Now let me begin to show you how retinal degeneration will look like in the zebrafish. And on this slide, I'm going to show you 2 pictures of the retinas prepared in a slightly different way. On your top left-hand corner, we have a normal control, which means that this is a normal, healthy retina from the zebrafish embryo on day 7. You see a lot of green blobs or green cells here. These are the rod photoreceptors of the normal retina. They are labeled by a green fluorescent protein. It's a long name, but basically this is a way for us to see these cells under the microscope. In the right preparation, these cells will emit green light. So again, on the top left-hand corner, we are seeing the rod photoreceptors in the little zebrafish embryo. More on the top, more at the bottom. On the top right-hand corner, I'm now showing you a night blindness fish with a name Q344X, which I will explain in a second. But the first thing that you can understand without knowing all the details is that these particular night blindness mutants, their retinas have far fewer rod photoreceptors. You only can see a few green cells in this retina. Why is that? Well, there is a long statement, and I will explain the important components also in the next slide. These mutants carry a human gene called rhodopsin. And this rhodopsin has a mutation called Q344X that are found in retinitis pigmentosa patients. So in human, the rhodopsin can mutate. And then one of the mutations is Q344X. And we will see this again, and I will explain very briefly what rhodopsin and the mutations are so that you can appreciate what you are seeing here. But the bottom line of this slide showing you a normal and night blindness retina is to tell you when fish carries the mutations that we find in human patients, their rods also degenerate, and substantially by day 7 in the mutant. So in the next picture, I'm going to give you a little more explanation what that crazy name rhodopsin is and what is the mutation about. This picture is a schematic diagram of the rod photoreceptor that has several different compartments. We don't need to know all of them, but what I am highlighting in the slide or the picture is I magnified a region at the center of the rod photoreceptor. And I also labeled a protein that is called the rhodopsin in green. Rhodopsin is a protein that is found inside our rod photoreceptors. Proteins are the little molecular machineries doing the work that our body needs inside our different cells. And in the rod photoreceptors, rhodopsin is responsible for picking up the light from the environment and initiate signal transduction that tells our brain that we see some light. In a healthy, normal rod photoreceptor, the rhodopsin protein would be produced and would be pushed out to a particular region of the rod photoreceptors called the rod outer segment, where the rhodopsin protein will be picking up the light and work with other proteins to tell ourselves that we see some light. When the rhodopsin protein carries a mutation like the Q344X that I told you, these particular mutation will affect how the cell can move the rhodopsin protein. Instead of moving the red mutated protein to the right place, they are stuck inside the cell. And consequently, the rod photoreceptors degenerate in the patients. The next slide I'm showing you the structure of the rhodopsin protein. Just to show you the location of the Q344. That name is actually referring to the building block position of the rhodopsin protein. Q is one of the amino acids, and it is found at the position 344. Rhodopsin is made of many different amino acids, and Q344 is one of the ones that can be mutated. In our example, it's mutated to an X, which means that it's a stop. So inside the cell, the rhodopsin with the mutation is a little shorter, and that creates all the problems that we briefly heard in the last few minutes. But the reason why I'm showing you this figure is to illustrate the context of the genetic problem that Ali also showed us a few minutes ago. There are many genes that can be mutated, and even in one gene, there can be many mutations. In the rhodopsin, all these red circles are the amino acids that have been found mutated in human patients, and they cause retinitis pigmentosa in different ways. And in fact, rhodopsin mutations constitute a big share of the retinitis pigmentosa inherited in the autosomal dominant form, as much as 25%. Autosomal dominant means like a way of inheritance. We all get our genes from our father and our mother. We get one gene from our father, one gene from our mother. And autosomal dominant means that as long as one of the two genes from our father or mother is mutated, we will get the disease. So I spent some time laying down the foundation to tell you why we like zebrafish as an eye disease research model. And I also tell you a few things about the retinitis pigmentosa model that we have been using in our laboratory. In the second part of the talk, I'm going to tell you one of the many ways that we can test the vision of the eye disease model and be able to find out the retinitis pigmentosa mutant in our laboratory. The way that I'm going to share with you today is shown on the next slide. And this arrangement is also telling you another advantage of the zebrafish model. The embryos that we collected from the zebrafish is actually are actually very small. The little black lines that you see in this particular picture are the little zebrafish embryo. They are just 2-3 millimeters long. So they are so small that we can put them in a small culturing container that we commonly use in the lab. This is called a multi-well plate. There are 96 wells or 96 containers in this particular plate that we can put 96 zebrafish embryos in the plate. In other words, when we do experiments, we can put the whole plate with 96 embryos into the machine and do 96 visual tests at the same time. So this speeds up our experiments a lot. And when we want to test the vision of these embryos, we put the whole plate inside the machine. And then we turn the light on and off. And in that sense, we are testing the light sensation of the embryo, capitalizing an inherent nature of the animal, because in the wild, in the river, like seeing a sudden light flash or seeing a big dark shadow is often a bad news. Like maybe the predators are coming and you better swim until you find a safe place. And so in other words, when we flash the little embryos with light or turn off the light, we startle the fish. They are scared and the fish will start to swim around, which we can capture by our camera. And you can also easily imagine if the fish has any retinal degeneration, they cannot see well, they cannot perceive the light change, and they will be somewhat indifferent to the testing situation, and they will not swim around like the normal fish. So I'm going to show you a video of such tests. In the next video, right now, we have 2 zebrafish embryo. They are sitting in the dark. Once I start to play the video, then we will see the 2 embryos. On the left, you have a normal one, and on the right, you have a retinal degeneration mutant. And you can see how they respond differently to a light flash. So I hope you can see that the normal one on the left, it doesn't like the light flash and it keeps swimming around actively for quite a while. Whereas the embryo on the right, which has retinal degeneration, it cannot detect the light change as much. So it doesn't really swim around like the normal counterpart. And in fact, we use the same arrangement to test the vision of the night blindness fish Q344X that I showed you earlier. And we extract the movement in terms of displacement on the y-axis on a plot. So the graphs shows you how much the embryos swim. When we turn off the light, in this case, at time 0, the black curve represents the normal wild type embryo. They are just 7 days old. When we turn off the light, the normal ones didn't like the light change. They swim around actively for 10 seconds or so. Whereas the Q344X night blindness embryos, they could not perceive the light change as well or as good as the normal counterparts, and they don't swim around. So we have a way to test the vision of the eye disease model that we have in the lab. And now that brings us to the last part of my talk, how we can use all the things that I have been telling you in the last 20 minutes or so to find new drugs that can potentially help the retinal degeneration patients. And that brings us back to the Multiwell plate that I showed you. That shows us another advantage of the fish model. And you can actually, again, imagine that fish are swimming in water. In these multiwell arrangements, the embryos are swimming in the water. So when we want to test drug, we can easily apply the drugs in the water that we contain the zebrafish. And the young embryos can absorb the drug. They can often absorb the drug through their skin. So in other words, testing drug in this animal is very straightforward, as simple as putting the drug in the water. We don't need to spend a lot of time feeding the animal with the drug. And in fact, we have used this setup to evaluate about 1,500 chemical compounds on the Q344X night blindness mutant. We are asking a very simple question. After we put a drug in the water, can the Q344X mutant perceive the light change better than before? If they perceive light change better than before, the drug may be helping the condition. And I'm going to show you one example in the next slide. It's coming from a drug from the screen called carvedilol. And I will tell you a few more things about this drug in a second. But what I would like to show you here is the plot coming from the light sensitive or light sensation test that I have been telling you in the last few minutes. And in fact, you have seen two of the curves before, the wild type and the Q344X. They are the same. When we turn off the light, the wild type doesn't like it. They swim around for a few seconds. And the Q344X, they don't see well. They don't care about the light change. The only difference here is that both wild type and the Q344X are treated with a solvent that we use to dissolve the drug carvedilol. And the key new result on this graph is the blue curve. The Q344X treated with carvedilol. The blue curve has a much higher activity than the red curve. That is the Q344X without the carvedilol treatment. So in other words, after the carvedilol treatment, the Q344X sense the light change better. The question is this treatment helping the rod photoreceptors? I'm showing you the same preparation that I briefly showed you at the beginning of the talk. On the top, we have a normal retina on the left-hand side and the Q344X night blindness mutant on the right-hand side with the rods largely degenerated. At the bottom, I'm showing you the same mutant after the carvedilol treatment. They have more rod photoreceptors. To us, this is an exciting news because there is no real treatment for retinitis pigmentosa that has rhodopsin mutation. And anything that can improve the situation a little better can potentially be a good news. So I'm going to begin to conclude. There are a few messages that I would like to share with you. I think our work is exciting and significant because if we find a drug that can help zebrafish with the rhodopsin, with the Q344X mutation, the drug may also help the night blindness patient with the same mutation. The other exciting thing about our study is that the carvedilol is also an FDA approved drug called CORAC. It's a beta blocker that has been used to treat high blood pressure and heart failure. Why is this exciting? If a drug is approved by the FDA for treating one disease, and in this case, this is the heart disease, developing and approving it for treating a different disease can be cheaper and faster because the drug is already tested in humans and deemed safe for human use. So if we do more research and find that the carvedilol is really applicable for retinitis pigmentosa, we can potentially bring it to clinical use in a much shorter time frame compared with developing a brand new drug. Therefore, in our screen, out of the 1,500 compounds that we screen, quite a lot of them are the FDA approved compounds with the idea that we want to speed up our discovery. To conclude, I'm going to share with you the current ongoing research directions in the lab. Of course, once we find a compound, we want to ask the question why it is helping the retinitis pigmentosa fish to have more cells or to see better. We are doing experiments to characterize the therapeutic mechanism. In order to bring that to clinic, we would like to test that in other animal models. For example, we have an ongoing collaboration with a mouse lab that has the Q344X mouse. So they are actually formulating the carvedilol in an eye drop format, and they are going to test that in the mouse model. If the drug works in both zebrafish and mouse model, then we are even more confident that the drug may be helpful in the human disease. And the final point that I'm going to share with you is that what we are doing now is not restricted to one particular eye disease or one particular mutation that causes eye disease. As long as we make a fish model that shows a problem in the screening assay, we are in business. We can potentially use that model to study disease mechanism and find new drugs in the way that I have been telling you in the last 30 minutes. And our goal in the long run is we are hoping to use our approach to identify more possible drug candidates that can be tested and potentially be brought to clinic to help patients suffering from different types of eye diseases. And I thank you for your attention, and I am going to stop here, and I'm happy to answer any questions that you may have. Thank you very much.
I had a quick question. I was actually curious on why you decided to use zebrafish instead of any other specific species or anything like that.
Could you repeat the first part of the question again? I didn't hear.
I just was curious on why you chose zebrafish as the test subject.
Oh, you mean like I myself?
Yes.
Okay, there are scientific reasons and non-scientific reasons. The scientific reasons, quite a few of those are the ones that I told you earlier in the talk, like they are small, they are cheap, and they are growing fast. So like in many ways with these advantages, we can do a lot of experiments cheaply and quickly inside the lab. And these are the scientific reasons that brought me into the zebrafish field. Like when I was in Hong Kong as a PhD student, I was actually doing human genetics, studying the mutations in patients. But I wanted an animal model that we can do experiments easily. So when I came to America for my postdoctoral study, I picked up the fish model. And that is the scientific part of the motivation. The non-scientific part is my original career plan was to go back to Hong Kong. And if you have ever heard about Hong Kong, it's a very expensive place, very crowded, and then and very expensive to live. And so I wanted to learn a cheaper animal model that we can keep cheaply in a small area so that I can continue my work in Hong Kong. But well, life always takes interesting turns, and then so instead of finding a job in Hong Kong, I found a job in America at Purdue, so I stayed.
Thank you very much.
I have a comment, not so much a question. May I do that?
Oh, yeah, please.
Yeah, I've heard about the zebrafish testing for a number of years, but I've never heard anybody break it down the way you have. And from somebody that has retinitis pigmentosa, it really shines a light on what you're doing. And I'm very thankful that you're taking that course of action. It appears to show great promise, and I'm very appreciative.
I'm glad that my students and my work can help, can ultimately help some people. Like we are, in many ways, like we should take the credit on the zebrafish, because like, they are telling us what the new directions can be. Sometimes like research is like this. We may understand the mechanism first to devise intelligently, like how can we fix the problem. But for a lot of clinical problems, sometimes like we identify the solution first and we figure out why it's working. And in the end, to me, who have been working in the medical research field for many years, the ultimate goal is like if we can find some ways to help the patients, we don't really care like how it was started. And then so to me, like this is a very interesting and good way, and we would like to continue to do that and do more using our professional time.
Many thanks.
Yes, doctor.
Yes, go ahead, please.
I'm fascinated with what you said. I too happened to read a little bit about the fish, but I appreciated how you broke that down. And I think of you know, like how soon might it start with mice?
Oh, my collaborator is currently actually making the eye drop with carvedilol. She has the mouse, she created that mouse. So we are working together right now, not only at the scientific level, but also at the like grant application level. We actually submitted a grant to the National Institutes of Health and hope they have mercy and give us some fund to continue our research. And we will, in our proposal, we want to use fish to understand why the drug is working in the fish model in that way. And we will also find out whether the drug is going to work in the mouse model. And again, like these are the foundation steps for us to begin to talk to other pharmaceutical professionals that we have something that can potentially be repurposed. Like that is the technical term in the drug discovery research. They repurpose an FDA approved compound for another use. So like we are collecting the necessary data to begin the conversation.
And the question is that once you get like the publish it, that in regards to that, then when you go from fish to mice, then to the human subject, do you have to then do the trials on human subjects, or does this drug help to support that it would be an effective tool?
Okay. Okay. What I can tell you is like first, I will say it in the safe and the diplomatic way. When we are talking about drug repurposing, the reason why a lot of people in the research field, or like everyone, is excited is that if we go through the clinical trial, the time is going to be short. Because a typical new drug development takes probably like as long as 17 years. And a substantial part is to make sure that you do multiple trials in humans, because you need to make sure that the drug is safe when you give to human. You need to test the toxicity, pharmacology, and all these things. And then when you repurpose an already approved drug, a lot of the safety profile is already known. The drug has already been tested in humans. So depending on the particular disease and the study, you may not need to go through that length in terms of human safety trial in order to go through the drug development process. Maybe like some people would, in some other examples, they would go directly into what we call like phase 2 or phase 3, which is like to test the drug efficacy. But like the other way, more courageous way to say about like repurposing FDA approved drug is that in theory, according to the regulations in the state, if a drug is approved for human use for one disease, in an emergency, the doctor can actually decide they can prescribe the drug off-label. So if they think there is a benefit and then the risk is minimal, they can choose to prescribe the drug even without going through whatever clinical trials. And so, but I cannot say for the doctors. I only know that this can be an option. So in that sense, using what I have told you from both angles, the bottom line is studying the existing FDA approved drugs can potentially bring treatments to the corresponding patients much faster than starting from scratch on new compounds.
Question then, say like, has there ever been a report where a person who had retinitis pigmentosa was taking that drug because of, what was it you said, heart condition?
Heart condition, yeah, like that is a thing that I actually talked to a student about. I think it requires some epidemiological study because heart disease is very, very common. And I think and Correct has been in the market for many years. So I think it would be a fun study. I tried to look whether there are published papers, but like a quick search didn't find anything. So I think like the right way is to do a deeper epidemiological study and then to investigate over the years among all those heart patients that have taken the drugs. Are there any ones that have retinitis pigmentosa? So like if we can extract all those information, maybe we can already see some correlation whether the drug has any clinical value. But of course, I should have a footnote here. Right now, our study is focused on one mutation on one particular protein that causes retinitis pigmentosa. Part of our research in the grant application that we submit to NIH is that we want to test the carvedilol, the Correct, also on other types of retinitis pigmentosa models that we have. Because if the drug can be generally applicable to different subtypes, then the utility would be even higher. And of course, like using your suggestion and idea, if we can look at the patient's data and collect all the retinitis pigmentosa who have ever taken Correct for heart disease, we may also see the pattern. And we are interested in doing that, but at the moment, I don't have the answer for this particular question. But you have got the right idea.
You mentioned about mutation, that there were 65 different type of mutations in retinitis RP.
In Ali's slide, she says 65 genes, but each gene can have many different mutations. So in the end, there are a lot of different types of retinitis pigmentosa that are caused by different genes and different mutations. And
I have a question.
Okay.
I'm sorry.
Oh, I'm only going. You go ahead first. Sorry.
Just regarding that, I also have dominant RP, but I have RP1. So,
okay.
You know, that you are very gene specific in your study, but with it being still a dominant gene, do you see this as just being a springboard for all dominant RP genes, or?
Right.
How do you?
Well, I think like we will have to do the experiments to find out, just like what I say a minute ago. We are going to use like different types of RP model and mutations to test whether the drug can potentially have effect in a general sense, or whether it is generally applicable. I'm somewhat agnostic because if you read a little bit more about the etiology and pathophysiology about retinal degeneration, or specifically RP, even autosomal dominant form and even mutations in rhodopsin have different subclasses. And through biological researchers, scientists have found that each class actually causes the cell to die in a slightly different way. So the reason why I'm saying agnostic is like if the mechanism is different, then it's probably more logical to think that a drug works in one class. Like Q344X is what we call class 1. And then it may not work on the other classes. But of course, I would also say agnostic in the sense that like very often, in the way that I described earlier when I answered Jim's question, is like sometimes when we find new treatments, we may not even know the mechanism. Maybe like a drug can hit like a common target and the sweet spot that is the weak spots of all subtypes of rhodopsin mutations. Then we can potentially have a generic drug that can be applicable to everyone. But as of now, I can't say either way because we haven't done the experiment. And the holy grail in the research field is really every time we find a drug, our colleagues are going to ask us, is this going to be generally applicable? And sometimes some like not so friendly ones, they say, oh, if it's not generally applicable, we are not interested in that. I have a neutral view on that because like to a particular group of patients, a treatment is a treatment is a treatment. So like a treatment is doesn't have to be general treatment to be valuable. And so by using FDA approved compounds as the starting point, we have a good chance of finding treatments for the specific subtypes, because we all know, I'm sure many of us know, like retinitis pigmentosa and some of the rarer form of retinal degenerations, they are called orphan disease. And the major scientific circle, they don't spend a lot of effort on that because like the patient pool on a relative scale is smaller. And then so we are hoping that like our way can potentially find some treatments like for these rare diseases one at a time.
Yeah, me too. Thank you so much for your work. I appreciate everything you're doing.
Thank you.
All right, I think we're ready to move on to the next speaker. Thank you so much, Bai.
Yeah, thank you.
That was a great presentation.
Thank you.
Vicki, let's go on to you to introduce our next speaker.
Okay, thank you again. That was so interesting. So our next speaker we'd like to introduce is Fred Fulkerson. He is the Senior Blind Services Coordinator at the League for the Blind and Disabled. He teaches independent living skills to people 55 and older who lose their vision. And he also provides information or resources to help them stay as independent as possible in their homes and communities. He has a bachelor's degree in interpersonal communication, and outside of work life, he likes to help others and take care of his family. He enjoys listening to and playing music and likes listening to comedy. So maybe he'll have a joke or two for us. Thank you.
All right, can you hear me?
Yes.
Yes.
Okay. All right. Like she was saying, my name is Fred Fulkerson, and I work with mostly seniors. So what happens is they start losing their vision, and their whole world really is upside down. They can't, or they think they can't do the same things they used to do. So it's my job to help encourage them as well, as teach them ways to accomplish everything they used to do, or as much of. The first thing a lot of them ask is, well, I want to drive again. Well, sorry, I can't help you with that one. That's one that I definitely can't help with. I'm totally blind myself, so everything that I teach and the technology and the gadgets that I show them, I use and have used since I was a kid. And of course, some of the technology, of course, has gotten a lot better, and some of it wasn't even around. So there's a lot of ways that don't involve technology that comes in really helpful, because seniors, some seniors aren't very tech savvy, and that comes in handy, that there's ways to do things without using technology. I didn't start using a lot of technology myself to help me with different vision issues until probably 15 years ago. Before then, I kept things pretty basic. So some of the things that teach people to use as well, they wonder how are they going to heat up their coffee, or heat up their lunch. And one of the most common things is putting markers or tactile dots on the face of the microwave, because they're all flat screen now, and you very rarely see one that's a turn dial. And so little sticky dots, or I call them sticky dots. They're called tactile dots. You stick them on the face of the microwave, and people are able to feel, or in some cases, for those who still have some vision to use, if you put different colored dots, they may see those dots stand out further on the microwave itself. Now, I'm going to hold up. It's going to be a little difficult for me because I can't see what the camera is seeing, but I'm holding up a sheet with different dots. There's different sizes of dots. There's different shapes. And the reason for that is because they can be indicators of different reference points, or different shaped dots to identify different products. The tactile dots you can stick on all kinds of items. If you're trying to differentiate the difference between shampoo and conditioner, you stick one on the cap of the shampoo. Now, if it's a sticky dot, it's not going to stay on there in the shower because it's wet, but you could glue it and reuse that cap. That's one technique of differentiating the difference and having it be a permanent solution, or at least for quite a while anyway. And that seems to solve a lot of problems. People can use their appliances that way, washers and dryers, dishwashers. I have them on my dishwasher at home. Let's see. A big thing people have problem, well, obviously, is reading. And then, of course, simple solutions to that are audio reading services for those who have any impairment of struggle with reading, all the way from dyslexia to, several other degrees of blindness. And so one of my parts of my job is to get people signed up with audio reading programs or audio reading service here in Fort Wayne area, which they have them across the country as well, which allows you to be able to listen to newspaper articles and different magazine articles and programming that they have volunteer readers read, and it goes out on air on a specific radio station. We usually have these magnifiers that are a little bit stronger than the ones that you can get in the stores. Ours are lighted. They have light on them. Holding it up. This one's kind of squarish on the top with a handle that points out kind of diagonal. It's a 3.5 magnifier. Our magnifiers start at like 3 times and go up to, I think 14 is the strongest one we have, which is really strong from what I could tell, from what other people tell me. So what we do is assessments on those, and we start at the low end, and we go up, and magnifiers can get too strong, and when they get too strong, they'll start clouding up, and people won't be able to see as good out of them, just like in the very weak ones. So we work our way up from the low end until we get too strong, and then we back off one, and then there's your magnifier that's going to be beneficial to you, the most beneficial to you. Let's see, other issues that are easily solved with tools that we have are talking products. We have several talking products. Matter of fact, the more and more these days, they have talking microwaves now, and talking toaster ovens and cooking appliances, liquid levelers, a little gadget that you hang on the side of the cup, and when the liquid gets to a certain point, it touches these prongs that are hanging on the inside of the cup, and it beeps at you. So them come in handy. Other things, learning to cook safely. There's different methods and tools for cooking, measuring cups that are specifically marked, with identification markers, techniques of having, say, a pan on your stove. You always keep the handle at the same spot. You have to feel a little bit more using gloves, oven mitts, long oven mitts, using your spatula to feel around a little bit more, or actually a lot more, I should say, to be able to feel where the edge of your pan is, where the handle's not, and then using that to find your food inside the pan, and so on and so forth. The other talking products, talking timers, people use them quite a bit. Some of the some of the other very common problems is as simple as knowing what time it is. So we have talking clocks. Talking clocks are a dime a dozen, and just as simple as this is just a basic talking clock, very cheap, like $15 clock, and it is 12:18 p.m. So that tells you, the time. It has an alarm on it. It has hourly chime if you want to do something like that. Talking blood pressure cuff. I'm showing that on the screen. I don't want to start it, if I get it started, it's gonna say all kinds of things and it's hard to shut back up. But it goes on your wrist, and it tells you your blood pressure and your pulse rate or your heart rate, and tells you if it's normal or if it's hypertension, or and comes in really handy. I use one of them. I use it myself. That's I use it quite often, actually. So and so as you can see, there it goes. He keeps talking. So there's other devices and other tools that we can use, labeling systems and everything, such as there's a gadget called the Pen Friend, which is a labeling system which allows you to label food. I'm holding it up to the screen. I have a Pen Friend in my hand. It's a gadget that is, I don't know, 7-8 inches long, maybe less. It's a little pen that you touch to magnetic discs or stickers that you can stick on things and record your own voice on each label, and it will let you identify using that method. There's also, the Pen Friend also comes with laundry labels, these tags that you can stick on your laundry to identify your clothing. Now, another thing that, never mind the technology, and one way to tell your laundry colors and things that match is sewing beads on the inside of your clothing. So you can tell what matches and what doesn't. Say triangle beads, the two triangle beads, one on the pants and one on the shirt, that pants and shirt go together. That's a very simple and easy task to do. You get them sewn on the inside of your clothes. There's several different sizes and shapes of beads and buttons and all types of things that you can get, at any hobby store or sewing department. Them come in really handy. And like I said, you don't have to have any kind of technology at all for that simple task. Let's see. Now, other technology, we have, people are wanting to know colors, so we have color identifiers. I'm holding another gadget up to the camera here, and you turn it on, and there's specific things you do to calibrate and all, but it's talking and it's beeping. I'll touch it to my shirt. It says it's blue. Now, if it's accurate, then that means my shirt's blue. I'm going to take its word for it because I have not asked what color my shirt was today. So that's another gadget. Now, a lot of these gadgets, they're kind of costly, which is not very fair to people with the disability, because a lot of people are on fixed incomes and have a trouble affording some of these, like the identifier. The color identifier is up to $200 or something like that. That's a little outrageous but it's really handy and can help you out quite a bit. Now, some of the other things we do is help people find resources, transportation. In Fort Wayne, we use the CityLink access bus, which is paratransit, and they have fixed route buses too, which you have that option. But for those who really can't make it to the bus stops and that type of thing, then they have paratransit, which is curb to curb service. So they will pick you up at your house. You schedule rides out, they pick you up at your house, take you where you want to go, and drop you off. I use it myself personally to get back and forth to work, and it's very helpful. It's another helpful tool. And let's see. Seniors and technology. Now, one thing that's really popular is smart speakers now these days. Seniors are really, because I work with a lot of seniors that are in their 70s and 80s, and they're not so open to technology, but they seem to have been a little more they've decided that some of these smart speakers are beneficial to them because they don't have to do anything. Once, son or daughter, grandkid or whatever sets them up with this, it's all a hands-free device. You don't have to do anything. All the person has to do is talk to the speaker with the trigger word, depending on what you have, the Alexas and the Googles, and there's a few other ones out there as well. And this takes away the need for several of the gadgets that I've been showing you, or a few different ones anyway, which is telling what time it is. You can ask the smart speaker what time it is, or set alarms, timers. You can set appointment reminders, medicine reminders. There's just, them are just some of the simple applications, never mind the checking the weather, reading audiobooks, all through a smart speaker. And also one that's really popular is making hands-free phone calls, because a problem seems to be really common is dialing a phone. It seems to be another common problem with so the smart speakers you can make phone calls. Some of them now have cameras, so the sons and daughters and their grandkids can have the same device in their house and call their loved one and be able to see if everything's okay, instead of worrying about, well, if I call grandma and she fell, she's not going to tell me. Well, if you got a camera there, you can see, that everything's okay or not okay. That's the way some of these smart speakers and everything are helping along. And like I said, you can bypass a lot of the gadgets. There's one thing smart speakers don't do is color identify, or identify your food, which kind of moves me on to more technology that is helpful, and that is smartphones and or computers. And smartphones, of course, are beneficial because you can use apps for all types of reasons: money identifiers, document readers. The way they make phones with the accessibility features on now these days, which is it's called VoiceOver for an iPhone and TalkBack for Android phones. And me personally, I use an iPhone for everything, quite a bit. And it has the personal assistant, just like a smart speaker, which, you know, you have Siri and then you have Google Instant Assistant and different voice assistants like that. There are other accessibility features for low vision. There's nice magnifiers that are built into the smartphones. Magnifiers, you can change the contrast, you can increase the fonts and the style of your icons, and which also computers, you know, have the same beneficial features as well. Now, smartphones aren't very popular with the seniors. They're still attached and used to their flip phones. So a common question is, where can I find a phone that's easy to use and has bigger buttons? One that I recommend, or I don't say I recommend, I suggest, is Jitterbug seems to have a lot of phones that are geared towards the seniors that the seniors seem to love a lot. And ones that once they my blood pressure machine is going off again. Once they get used to, if you can get them to get used to the smartphone, it allows them to have the ability to be able to talk to their phone and have them call people at random, you know, whoever's in their contact list, or they can rattle off a phone number and have it call that way. But like I said, that's one of the harder things for people in their 80s. It seems to be to get grasp on. Not so much people in their 60s, which, you know, there seems to be cutoff points there, you know, for age wise, about what technology is acceptable and what's not. Now, some of the other things that would fall under the category of senior blind services is peer support, in which I know for sure there's Patrick that's listening, and he's a peer support coordinator. And these groups are just what they sound like, and it's support groups. Yeah. And they are for people to help each other out and learn different topics and subject matter that might help them solve problems. You know, one person who is blind talking to another person who is blind are going to be able to help each other figure out different ways to do what they're set out to accomplish. And sometimes encouragement. One of my mottos that I use at work is, when I go to see a consumer, I want to leave them feeling better than they did when I got there. So I want them to feel more encouraged when I leave than they did when I got there. It works most of the time. And I work several support groups in the outerlying areas because the league works in several different counties, so we reach out to quite a bit. I work 9 counties and then I have a counterpart that works 5 counties out further than that, further than I go. So all the way out to the South Bend area, St. Joe and everything. So we get out there pretty far. Now, specifically, my program is senior blind services, but we also have somebody who does the same work with people under 55. And all the same things apply, and except for the technology isn't so hard to sell the idea of with kids these days start young. So you can hand a 2-year-old a smartphone and they're all over YouTube. So computers the same way. Now computers, I use a computer all the time with a screen reader. My screen reader reads what's on the screen. I don't use a mouse. I use the cursor keys to get to where I need to go on the computer, and it works well. I use it, and I use between a smartphone and a computer. I use it every day to do my work. I mean, it's invaluable pieces of tools that I get to access the information that I need. And nowadays the computers have such good accessibility features for anyone who starts losing their vision to totally blind.
Hey, Fred.
Yes.
This is Allie. I'm hoping we can hold the Q&A for your presentation. I think I mentioned Anthony Ferraro is the motivational speaker for today, and he's on a crunch line because he's I believe he's at a performing musically. So I want to make sure that he's on right now, and that once he goes off, whether we can continue your presentation or just answer questions. I just want to make sure that we get everyone a part of this.
Yeah, that's not a problem.
Are you sure?
Absolutely.
Okay. So to give a quick introduction to Anthony, he was born with Leber's congenital amaurosis, and he's a motivational speaker. He's a athlete, musician. So I'm very happy to introduce him real quickly. And once again, thank you so much for joining in the middle of your your break.
Can you hear me?
Yes.
Awesome. Thank you so much for having me. Yeah, I'm actually on the break I'm playing a gig right now, playing guitar, and I'm so excited to be here. Thank you for having me. Gonna give you guys like a short, overview of like my life and my story. So, I'm Anthony Ferraro. I'm 26 years old, and I was born blind. I had a little bit of vision when I was younger, and I grew up in a small beach town with like 60 cousins. I was the youngest of 5. My mom was the second oldest of 13. So I had all these people around me all the time, and I was always like kind of never treated any differently, and they were doing all these things around me, so I wanted to as well. So I learned how to skateboard and surf, and like I used to ride bikes until I started hitting parked cars, and then my mom was like, Anthony, time to get off the bike. You got to at least get on the back of like a tandem or something. So like there's always been ways that through my life that I've like made adaptations and figured out ways to make things either accessible or just doable for me, and not letting like blindness or something.
Did we lose him? I hope he. Oh, there we go. We got him on video. Wonderful. Hello.
Hello?
Yes, we can hear you. Perfect.
Okay, cool. Sorry. Where did you lose me?
Uh, about skateboarding, hitting cars, around there.
All right, cool. So I like I grew up skateboarding and surfing and like doing all these things, you know, making like figuring out adaptations and like just wanting to do certain things and not letting my like blindness hold me back and just figuring out ways. And um I went to a school for the blind and visually impaired. Uh I went to a school for the blind and visually impaired in Philadelphia. I would commute every day from Spring Lake to Philly, and it'd be a 2-hour commute. And that's where I learned how to like read and write Braille, and you know, basically learn everything I needed to integrate into the visual world of school and things like that. And after that, I transferred to the public school in my town, and I started going to school with sighted people, you know, every day. And I went from being the only blind person the to being with all blind people to being the only blind person. And I used to like be really insecure and like not comfortable with myself and try to like never use my cane and you know like hide my braille books and stuff like under my desk, like all these things. And I just like I found I found that like once I accepted that and like once I accepted my blindness and was able to use the cane and like, you know, learn these tools around me, because there's so many different tools to make life easier. Like it made life easier because I was dealing with all this anxiety about like being, you know, scared of like people finding out I was blind, all this like negative stuff, and it wasn't real. And I started wrestling in 7th grade and I did terrible. I was so bad. Like I went 2-12. One of my wins was a forfeit where I just went out and got my hand raised. And then I at the end of that season, I was like, you know, I said to my dad, I need to get good at this, Dad. Like I want to get good at wrestling. I don't want to just be like mediocre, blah, blah, blah. And he took me to a wrestling club and I started going 4 nights a week and then to tournaments all around like New Jersey, New York, and Connecticut and Pennsylvania, like all around for like every weekend that I could. We would go on these trips to tournaments, and it was so demoralizing because I would get beat every day, like every tournament. I wouldn't even score a point, and all I wanted to do was win. And I remember leaving these tournaments like so upset, you know, like why can't I win? Like why can't I win? And I started making my goal a little smaller, you know, instead of winning a match, I'm like, okay, let me score a point. And I started like working towards that to get to winning a match. And I started scoring points and like seeing these little, you know, advancements after putting this time in. And I started winning matches finally. And then in 8th grade, I ended up winning the whole Tri-County championship. And one thing I experienced in wrestling was a lot of adversity because people didn't like the fact that I was blind and they had to adapt to like my wrestling style. So because I wrestled sighted people, they had to stay in constant contact. And people would say things to me like, you have an unfair advantage, or you're faking your blindness, all these different things, because I was like getting good and winning matches. And I had to deal with that. You know, it was really tough. It was a struggle at first. And I just remember also in 8th grade, I was supposed to go to the high school my older brothers went to. And I had a handwritten letter of acceptance. And then I got a letter in 8th grade saying, you're no longer accepted to the school. We don't want to have to provide accommodations, you know, different resources like and things like that. And you won't fit into the culture. It was all this like really hurtful things. And I remember just being like 14 years old and being like, you know, this isn't fair, like yelling and being so angry. I went home and I was like crying, screaming, like so angry at the world, like why could this happen? And finally I just like kind of put my frustration into wrestling and like kept wrestling and putting all this time in and kind of a positive outlet to let it out. And I ended up going to another school, and I ended up going to another high school, and I ended up doing really well for myself with like wrestling, and I didn't know a single person going there, so it was really scary. And like Sorry about that. And I didn't know a single person going there, and I just like made it work. I remember just making friends, like one goal, make one friend, talk, like find someone to talk to and get out of my comfort zone. And I became a wrestling captain there for 3 years and started doing, I did really well. And my older brother, Oliver, was a filmer at the time, ended up teaming up with this guy to make a documentary about my life as a blind wrestler and growing up blind, dealing with adversity and different things. And they followed me around my entire senior year with these cameras and like filmed so much stuff in real time, like nothing scripted. And after my senior year, it got put on the shelf. And 2 years later, the guy Chris Sikorsky, who teamed up with my brother, took it off and said, there's an amazing story here. I have to put it together. Like, he won 122 matches. Like, he won championships in high school and stuff, and just the adversity and all that stuff. And he put together the first 15 minutes, and he loved what he had. And the day before my brother and him were supposed to meet to show my brother the film, the first 15 minutes of it, he passed away, my brother, at the age of 27, and he never got to see any of his film he created for me. And it was this like crazy impact, my family, the world. And after that, Chris vowed to us that he would finish the film, and he did exactly that. We raised all this money on Kickstarter, and the trailer got all these views. And someone reached one day, I'm sitting home, and someone reaches out and they're like, "Is this Anthony Ferraro? This is so and so from the United States Olympic Committee. Would you consider training judo for the Paralympics?" And wrestling's no longer in the Paralympics, but would you consider doing judo, which is very similar? And I was like, absolutely. Like, they asked if I would consider like learning this whole sport and going around the country and stuff and around the world to train and compete. And I packed like 10 years of judo into like 4 years. And for the past 5 years, that's what I've been doing. I've been training for the Paralympics in judo. I was like very, very close to making it to Tokyo, but I tore my groin right before the last two qualifying tournaments. I was number 1 in the country and like top 20 in the world. And then I got hurt. So now just like life, like things happen, things get in your way and like obstacles. I'm just going to keep going and keep working hard until 2024 for Paris to go to the Olympics. So I'm working really hard at that. And also on the side, I do music where I play gigs and play a guitar and sing for restaurants and bars and stuff. And that's how I make some of my living. And I also do motivational speaking. And, I make a lot of short form video content and like just try to inspire others and motivate others to like, whatever your situation is, whatever you're dealing with, you can get over. Like you can accomplish big things in life. Like you can dream big and actually work towards that. And, I tell people that the only disability is a bad attitude in life. So whatever you're doing, whatever things you might have that are hindering you, or you may think, it's like, use that to your advantage and start like getting out there and getting out of your comfort zone. And just whatever it is in life you want to do, if say you want to learn to sew, like go sew every day, because you're not going to just get good at that overnight. If you want to, skateboard, it takes time. Anything you want to do in life, like it takes hours, and it takes falls and failure, and it takes getting back up. But the most important thing is that you get back up more times than you fall. That's some of my story, and I really hope it resonates with some of you guys. And please know I'm always here as a resource. If you ever need to reach out to me, you can reach out to me on like Instagram and Facebook and all that stuff. And my website is asfvision.com, and you can email me. It's a@asfvision.com. And just anything you guys ever need, any questions or advice, or you just need someone to talk to, I'm always here. And I really appreciate you having me today.
Of course. Thank you for sharing that.
Thank you.
Since hopefully I think the hard stop is 5 minutes or so. Does anyone have any questions for Anthony?
Space. Anthony, I'd like to ask if it's okay if I use that quote, "The only disability is a bad attitude." I love it.
Please do.
Thank you.
Thank you.
One thing I actually have to ask is, given you do so much, athletes, motivational speaker, musician, do you have also almost married? So congratulations.
Yeah, thank you.
What is your main focus right now? Like, is just everything? Or
Taking
what?
life one day at a time.
Okay.
So right now, my main focus is getting married a week from today, and then my focus is I have to do some skateboarding things. I just making this video content creation takes up a lot of stuff, and then it's going to be a lot of training for the Olympics, like hardcore when I get home.
Anthony, thank you for sharing your story. It's very inspiring. And
thank you.
Congratulations to you on your upcoming nuptials, and good luck with Paralympics. That sounds awesome.
Thank you so much.
So I'm really not trying to promote anyone else, but if you have not, so I watched A Shot in the Dark. That's his documentary. Or just look up Anthony Ferraro. He has many viral videos. It goes into his wrestling career, as well as his brother, who was a part of that video, just passing away unexpectedly. It's very heartwarming.
Thank you so much.
Yeah, of course. Thank you so much for joining today. Where are you playing again right now?
This is a different gig for me. My favorite little organic supermarket, and they asked me to play on Customer Appreciation Day, and then tonight I have to go play a private party from 6:30 to 9:30 or something.
All right.
Anthony, what is your genre of music you play?
Blues, rock, country, folk, jazz, finger picking, a little bit of everything. I'm a man of the blues, though. That's my favorite.
Sounds good. Do you have videos of it?
Yeah, it's all on my YouTube. Me and my fiance did 2 cross-country tours where she drove from here to New Jersey to California and back, and then one to New Jersey and Denver, Colorado and back, and we stopped along the way in all these locations and set up with beautiful backdrops in different places and live streamed it to the world. And we did that during the pandemic, so we tried to spread positivity and get people happy during the time.
I have one quick question, Anthony. One of my favorite videos of you is when you're making hot dogs on the grill. It's fantastic. I was curious though, how many times did you actually end up burning yourself doing it? Because I know a grill can get very hot.
Quick fingers.
Okay.
Probably 20.
Okay.
Give or take.
Thank you. That was awesome.
Of course. No, thank you.
All right. Is that all of our questions for Anthony? Once again, this is being recorded, so we'll make sure to share this with everyone, but we want to make sure to use everyone's time wisely.
And please feel free, if you didn't have a question now and you have one later, just send me a message.
I will send an email not only with the recording to everyone who's registered, so not necessarily everyone on the call today, but to the recording, but as well as just contact information for each of our
Absolutely.
speakers.
Everything you can find on my website, all my contacts are on my website, which is just asfvision.com.
I'll make sure to include that.
Thank you so much.
No.
I really appreciate you having me.
Of course. I hope you have a good rest of your music.
Thank you so much. I hope you guys have a great day.
All right. Bye, Anthony.
Bye.
Well, as I mentioned, we are going to have a door prize drawing of a $50 Target gift card, but I really want to see if anyone has any questions for Fred Fulkerson. And like I said, I apologize if that seemed to cutting you off by any means. I just wanted to make sure someone on that deadline could speak.
I have a question for Fred. There's a couple companies out there, IRA and Be My Eyes. Do you find that useful for any of the clients that you have?
A lot of people, I've talked to a lot of people use Be My Eyes quite a bit. That seems to be pretty useful. The other service, I haven't heard a lot of people use it. Just occasionally I hear somebody talk about it.
It's expensive. The others, IRA's
Yeah.
fairly expensive.
Yeah, Be My Eyes is free, so
Right.
people tend to, you know, go towards that way.
How about Seeing AI? I have that one on my phone. Not that I've used it, but I just have
I
heard about
use it. I use it quite often, and
Okay.
I do tell people because it has so many different features. You know, it has the document scanner, money identifier, a light detector, a barcode scanner. You know, on and on. It's a very useful app.
What's the name of it again?
Seeing AI, like SEEING, and then AI.
Okay, hadn't heard of that one.
I'm not sure. I know it's there for the iPhone, and they were supposed to be coming out with an app that would work on an Android, and I don't know if it's out yet or not.
So Jim, if you have an iPhone, it's available. But sorry, Grace, what were you saying?
Yeah, Fred, I have a question. Do you ever get out to Goshen, Indiana, to Greencroft? At Greencroft, I'm going to be moving there in about November it's a retirement center community, and I know quite a few people have macular degeneration, and I'm the one that has RP. And I'm interested in forming a self-help group there. I thrive on self-help because thanks to Southeast Asia, they help me to mobilize self-help groups. And just wanted to know if you're aware of Greencroft, and have you been there? And maybe it sounds like we could have people like you or whoever to come and share. If we, like for instance, because they support that idea. They have a Parkinson's support group, but they said to me, they said, you know, hey Grace, that would be great to get that organized.
I have a counterpart that I was talking about that works Goshen area, because that's a couple hours away from Fort Wayne. But
right,
yeah, her name is Donna Horner. I don't have her contact information. I guess let's see. She would be the one that would work that area, and I know there are she does have low vision support groups around. And yeah, I think there's always room for more support groups. That's for sure. But she
right.
would be the senior blind services coordinator out in the Goshen area.
Fred, her email address would be donnah.h@the-league.org.
Ah, thanks, Patrick.
Allie, could you put that in? I don't know how to use that here when you put that, because that would be nice to know.
Yeah, I can email you separately with that, confirm with Fred and possibly with Patrick that email address.
Absolutely.
Okay. Perfect. That's all the questions for Fred for now. All right. So I probably overdid this, but I put together a virtual wheel of everyone who registered, which we had 30 people. Obviously not everyone's on the call, so we'll keep spinning the wheel until someone who's on the call can accept the $50 Target gift card. So let me share my screen. It's just a fun little thing we can. Okay. All right. So we might need to like spin it a couple times, but hopefully not. I actually think Luke is on the call. I'm going to stop sharing my screen. Luke, I believe I saw your name. I'm going to ask him to unmute. Because if he doesn't say anything, it doesn't count. Luke. Well, either way, I think that's fair enough. I see his name that he's present. But does anyone have any, like, just in terms of, I know we just have a minute left, but just wanting to be a part of the chapters program, that's kind of my spiel. If you enjoyed what we said today, please reach out to me. We want to plan these at least seasonally, and have a bunch of other programs, including the Vision Walk and just our national chapter vision webinars. So thank you all so much for joining. There is a recording, and like I said, if you want to become more involved with the Foundation Fighting Blindness, please reach out to me. I believe you all received an email from chapters at Fighting Blindness. So if you just respond to that email or respond back just saying, hey, I'm interested, I will receive that as well. But
Allie, I just wanted to say something real quick.
Yeah.
I wanted to thank you for organizing this whole chapter meeting. I thought it was very, very well done. And I want to thank all the speakers who participated, because like I said earlier, this means the world to me, and I'm glad that this information is being shared with everybody. So thank you very much.
I agree. Yes, all the presenters were wonderful. Thank you, Phi. Thank you, Fred. Thank you, Anthony, who's no longer on the call, but this was wonderful. This was our first virtual speaker series in however long, I can't remember. So thank you so much. No one has any questions? I want to make sure that no one feels like this is a one-way conversation by any means.
Thank you for allowing me to participate.
Of course. Thank you, Patrick.
Thanks, Patrick.
Well done, Allie.
Yes, well done. And I appreciate knowing about the group. Very helpful.
Of course, yeah. And so next week, probably, I don't know, Wednesday, you'll get a recording, but I want to make sure that we also provide that contact information. So Phi, Fred, I'll probably annoy you about asking what you'd like me to include in that.
Oh, not a problem. Thanks for
okay.
having me.
Of course.
And I would like to get Dr. Leung from Hong Kong, his address, because I've been to Hong Kong 4 times and knows the disability group, and we might know some people.
Oh yeah, well, Hong Kong, like don't get me started.
Yeah, I don't know if he's
about
going
Hong Kong.
And I was with the hearing impaired group and the blind community.
Wow, yeah, I noticed quite a few groups, but like yeah.
Okay,
Great.
but I would like to have your contact information so I can follow up with that.
Okay.
Yes, Grace and
Plus the
everyone, you'll
RP.
receive it. Yes.
Yeah.
You'll receive the presentation, you'll receive contact information for each of the presenters. And once again, thank you so much for joining us on your Saturday. This was a great meeting.
Thanks, everybody.
Thanks.
Bye.
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 Bye.